So good afternoon, thanks for coming. So my name is Veronika and it's my pleasure today to talk about the genomic basis of soil adaptation in the alpine carnation, Dianthus sylvestris, and about processes which facilitated that adaptation. So first of all, why it is even relevant to study soil adaptation? It's because soil imposes strong selection on plants, for example, through differences in pH, which basically determining availability of the nutrients, and also nutrient availability and physical properties of the soils act as a selective agents. And as we all know, plants are sessile, so they have to adapt if they want to survive. So they adapt through changes in physiological processes and also, for example, by selective uptake and transport of the nutrients. And importantly, the mosaic of the different soil types in the landscape contributes to the diversification in the biotic world. And here I just pick an example from a small region into Central Europe, from the European Alps. And even in a small scale of a few kilometers, you can see quite heterogeneous landscape here denoted by the differences in the soil pH. So the question is, what is the genomic basis of the soil adaptation? An important mechanism of adaptive response can be pleiotropy. It means that one gene affects multiple traits. So therefore, pleiotropy is a key mechanism mediating trait co-inheritance. And the traditional view on pleiotropy is actually that it has negative effects on adaptation, imposing cost of the complexity. But on the other hand, it can also have a positive impact, for example, through increased effect sizes. And pleiotropic mutations can be favored when populations are far from the optimum. And such case can occur during the range expansions when populations actually colonizing and adapting to the new environmental conditions. And according to the theory, adaptive pleiotropic genes are more connected and central within the gene networks than the other genes. So the question is, what is the impact on adaptation? And what is also important for adaptation is maintaining a high genetic diversity, especially when populations adapting to the new environments, such as during colonization and range expansions. And indeed, those range expansions can lead to the loss of the genetic diversity through a series of the founder effects. So the question is, what is the impact of that genetic loss on adaptation? And so to study these processes, we selected a model species, perennial carnation, Dianthus sylvestris, which occurs in the European Alps. So at the map, you can see locations of our 43 populations across the Alps. For which we have low coverage data available, so we mainly work with the population allele frequencies, and also for each of those populations, we have available the soil data, including pH measurements and also exchangeable ion concentration of 9 elements. And a short window to the demographic history. During the last glacial maximum, the Alps were covered by the ice. So populations had to retreat to the glacial refugia. And in the case of Dianthus sylvestris, those refugia were located in the southeastern Alps. And from that region, later on in the post-glacial period, populations expanded, recolonized the Alps. And as you can see from the PCA, population structure basically mirrors the geography in the Alps. So in the dash ellipse, we have the population from the former glacial refugia, and then on the left, we have the most western populations. And here I color the populations according to the similarity in soil. So you can really see a mosaic of the different soil types across the Alps. Okay, so I'm going to ask those three questions. So first of all, what is the genomic basis of adaptation to the different soils along the range expansion gradient in Dianthus sylvestris? So to explore the genomic basis of adaptation, we applied two approaches: environmental association analysis, LFMM, and then generalized dissimilarity modeling. So first of all, by environmental association analysis, we were searching for significant associations between the population allele frequencies and soil elemental concentrations. For each of the elemental concentration, we applied a univariate model. We identified almost 4,000 SNPs. And then to further refine the list of the candidate SNPs, we applied the second approach, generalized dissimilarity modeling. So for each candidate SNPs, we basically modeled the changes in the allelic turnover along the soil gradient of particle element. And importantly, here by the allelic turnover, I mean changes in genetic distances between any pair of the studied populations along the gradient. And here I pick a schematic example of output from GDM. So here it's just for one candidate SNP. So on the x-axis, we have the soil gradient, on the left side with low values, on the right side with the high values. And then on the y-axis, we have the allelic turnover. And we can check two things. So first of all, the height of the curve, which basically showing the magnitude of the allelic turnover, indicating importance of the particle SNP for adaptation. And then we can also check the shape of the fitted spline, which basically indicating the different selection along the gradient. And here we can see quite steep selection, steep curve towards the high values, indicating a stronger selection in those conditions. And then we found out that actually the highest magnitude of allelic turnover was observed along potassium, aluminium, and magnesium salt gradients. And here I pick an example of the aluminium-associated SNPs. And you can see the highest magnitude of allelic turnover and also the strongest selection at the high aluminium values, which makes sense because aluminium can be highly toxic for plants in acidic soils. Okay, so then we annotated those SNPs to the candidate genes. Here we basically work with the Arabidopsis thaliana orthodox, and we found candidate genes including mainly ion transporters of macronutrients and metals involved in organic anion transport, metal, ion homeostasis, calcium, ion transmembrane transport. And here I picked the variation specifically in the nitrate transporter. In our analysis, it was associated with another macronutrient, with calcium. And we can see that it's at high allele frequencies, mainly in populations coming from low content of calcium, and also from from the output from the GDM, the spline indicate basically the strongest selection in low calcium values. Okay, so now let's turn to the second question. If the pleiotropy played a role in facilitating adaptation to the different soil types. So we use connectivity and centrality in gene networks as a proxy for pleiotropy. And we found out that the candidate genes have more interactions than the other genes in the co-expression networks. So by the dashed red line, you can see the mean number of the gene interactions for the candidate genes, which is higher than mean number for randomly picked genes. And then we found the similar pattern for the centrality statistics. So we found out that actually candidate genes are more centered than the other genes in the gene co-expression networks. And here I picked the example of the one statistical node degree. Okay, so now let's turn to the last question. If the loss of the genetic diversity during the range expansion had a negative effect on soil adaptation. So we explore the genetic diversity of our populations for two categories of the SNPs, so for candidate SNPs and for the genome-wide SNPs. We calculated the observed heterozygosity, and you can see that in both cases we actually see the decrease in genetic diversity along the range expansion, so from east towards west. But for the candidate genes, they actually show an increased diversity. So this can mean that actually selection acting on adaptive variants contributes to the maintenance of the genetic diversity in populations. Okay, and so let me conclude. So I show you that populations responded to selection imposed by soil differences through polygenic adaptation involving ion transport and homeostasis. Then the differences in concentration of aluminium, potassium, and magnesium in soil caused the highest allelic turnovers. Then pleiotropic genes facilitated adaptations to new soil conditions. And loss of the genetic diversity during range expansion actually did not preclude adaptation at expansion front. And by that, I would like to thank to my collaborators and colleagues and you for your attention. Yeah, and I'm happy to take any questions.
So I thought your centrality statistics part was really cool. Why do you think that more pleiotropic your candidate genes are more pleiotropic in general? Like, do you think it's because it's polygenic adaptation, or what are your thoughts on that?
Yeah, it can be definitely that, and then I think it's really related to the history. So during the range expansion, actually populations have to quickly adapt to the new environmental conditions which are new to those populations. So this trait co-inheritance can be really important for facilitating this, like a rapid adaptation. Yes.
Like a little more about your last point. Yeah. PC1 regress with this one. How did you use this graph to approve your point?
Yeah, okay, so basically, as I mentioned at the beginning, that from the PC based on the genetic data, that it mirrors the geographic, so then I actually picked the principal components for each population, and then I aligned them in those two graphs. And then I calculated the observed heterozygosity for populations based on the genome-wide SNPs, and then based on the candidate SNPs, and then did the Wilcoxon run summary statistics, and actually found out that the diversity based on the candidate SNPs of the populations is significantly higher than the diversity of the populations based on the genome-wide SNPs.
Are these populations only the from populations?
No, all of them. So each point is one population. So we have 43 dots, 43 populations. Yeah. Okay.
Hi, everyone. I'm Joanna Rifkin. I'm at the Hudson Alpha Institute for Biotechnology, and I'm going to be telling you today about, oh, there is a pointer. Wonderful, wonderful. It's a better pointer. Great. I'm going to be talking to you today about drought adaptation in sorghum. Before I get started, I just want to spotlight the many funding sources that go into a really big project like this, and the many people who work on a big project like this. This is a very it-takes-a-village kind of project. I particularly want to spotlight Jesse Lasky, Avril Harder, and Avinash Sridasayam, all of whose work features prominently in this talk. The pointer has stopped working. So climate change is a major threat to many aspects of life in the next century, particularly including agriculture. These plots are showing a couple of models of the increase in extreme drought by the end of the century we're living in now. Even the optimistic outlook is not good. So we need to be developing drought-adapted and drought-resilient crops to continue to survive as a species that eats food. Sorghum is a really promising arena to work on for this because it's already got a lot of circulating drought tolerance. It evolved in a very dry place, and there's a lot of variation within it for drought tolerance. It's used for many functions, including as a staple crop. And what you can see from this plot is that in the center we have a non-drought tolerant line, and along the sides we have drought tolerant lines, and that's just existing circulating variation that is known in the species. Very sad, this point of situation. Before you go do a big project like this, you need to think carefully about what lines you're putting out into the field, because phenotyping on this scale is extremely demanding. Agriculturally relevant experiments have thousands, tens of thousands, sometimes hundreds of thousands of plants in them. They're phenotyped often with drones. It's very hard to translate work in the lab to actually efficacy in the field. So narrowing the kinds of variation that we do these large-scale tests on can improve our odds of success. One way to look at this is selection scans, which are a tool from evolutionary biology. If we can identify allelic variation that correlates to environmental variation, like the excellent talk we just saw on carnations, that can help us decide what kinds of lines and varieties we want to test in the field. If, for example, we wanted to breed Arabidopsis with better flowering times, we might consider looking at variants that held this SNP from the dog1 gene. Sorghum is a great system for this because it's really diverse. It was domesticated somewhere around here. I don't even have a pointer. This is tragic. It was domesticated somewhere around Ethiopia 4,000 or 5,000 years ago, and you can see from the picture in the center that there's still a lot of domestication in its original range. You can also see that it spread with human migrations, came back into secondary contact with itself, went through bottlenecks as it was transported to the south. So it's got a complex history, but that holds a lot of diversity within it. Sorghum is a great species for this. The sorghum pangenome is a great resource to work on this with. The JGI is releasing now this new resource, including 30 separately assembled reference genomes. There have been over 10,000 lines genotyped to some degree, including over 2,000 that have been fully resequenced. My work today is mostly going to focus, however, on 433 land races. So these are varieties that were developed by farmers to meet their needs. They haven't been through major crop breeding bottlenecks. Instead, they're still holding a lot of variation from the original variation of the species. So I've told you a little bit about what sorghum is and why this matters for climate change. I'm now going to tell you a bit about the population genetics and about signatures of selection in sorghum. So these are just the locations of the land races we're working with. There's an inset so you can see in detail. I've just grouped them geographically. This is just partitioning around medoids, so around those little black asterisks, weighting by latitude and longitude. It's just dividing them up in space. What I really want you to notice here is in the inset you can see maybe there's triangles and circles. The triangles, according to our best climate models, do not experience drought stress during their growing period. The circles do. So the triangle-circle milieu, it's really closely mixed. There's triangles and circles right next to each other. So we think that we're getting climate variation across the range. And we know we're capturing a lot of genetic variation. These are the 10 chromosomes of sorghum, pi, nucleotide diversity on the y-axis. I draw your attention particularly to the thick black line on each of these chromosomes. That's nucleotide diversity in national breeding programs. So the breeding programs have been selecting, they've also been bottlenecking, and you can see that the land races in all the pretty colors are retaining diversity that is not in the breeding populations, and that there's a lot of variation in where the variation is across the genome. So ideally, we take this wonderful genetic variation, we take this wonderful geographic variation, we identify promising SNPs, and then we give them to farmers to do experiments with. Great. But that's really hard. And we very often rely on approaches that discretize our population genetics, so that draw lines and say, we're comparing this population to this population, and we introduce a huge amount of uncertainty when we are drawing those lines and making those discrete groupings. We have to decide where to delineate subpopulations, and we might be capturing the wrong scales. We might be capturing the wrong scales in the wrong places. We might be conflating ancestry with adaptation. One way to try to correct for some of this, and this is very much drawing on Jesse Lasky's work, please bother him about the actual math, is to use more continuous mathematical approaches. So I'm showing you here allele frequency variation at two different scales. At the top, we have an allele that is turning over a lot. Thanks, Veronika, for introducing the idea of allelic turnover. I was worried about that. And at the bottom, we have a SNP that is turning over slowly. And rather than trying to draw lines and compare allele frequencies between blocks, we can model this using continuous math drawn from the math that people use to describe waves. This is a wavelet transformation. Physicists love wave math. You can use it to describe music. Brownie points to anyone who recognizes the music. But it also turns out to work really well for describing allele frequencies. So at the top and bottom, we have wavelet transforms of these allele frequencies varying in space at two different spatial scales, a small one and a large one. And this really cool paper that just came out that I've basically been beta testing offers approaches to model allele frequency with wavelets across different scales. So what does that look like in practice? I'm going to apply this to two questions. How does the scale of turnover vary with drought? And what types of loci show extreme turnover? So I'm just going to show you a graph here. On the y-axis, we have wavelet dissimilarity. That's how dissimilar populations are from each other in allele frequencies on average on different scales. The gray-blue polygon, that's what it would be if at every scale things were just entirely randomly permuted. We shake up our populations at each scale. They're randomly positioned. So up to 100 kilometers, they're more similar to their neighbors than random. That's a fairly high scale to be so similar. In Arabidopsis, that crosses the line at about 25, because Arabidopsis just sits. These, however, humans are moving them around, they're interbreeding, and above 100 kilometers, the turnover increases. We can also look at how this varies in space. It's not consistent, it's really heterogeneous. So if you think of that circle on the left as a sampling radius around a population, if it's clear, it's effectively panmictic with its neighbors in that range. The more red it is, the more different it is. So we're seeing a lot of variation in how much allele frequency turnover there is in different places in space. How does this relate to drought? Really interestingly, things that are exposed to drought are more similar than non-drought exposed populations. So somehow there's more similarity in allele frequency over longer distances for drought exposed populations. Okay, so that's cool. Can we identify loci that are important to this? So the way what I'm going to show you next works is we've modeled our loci with this wavelet transform. We're now looking for extreme allele frequency turnover at different scales. We have the scales in different colors. We have our chromosome 7 on the x-axis, and on the y-axis we have a negative log transform of significance. Prepare for some sprinkles. This is obviously a needle in a haystack. There's different loci that pop out at different scales, and as in many genomics approaches, the challenge is actually getting the needle out of the haystack. One approach to this is to look at gene ontology enrichments. So to say, okay, our top 10,000 hits across the genome that are in genes, what kinds of genes are they in? I'm drawing your attention to this peak now. We're going to zoom in on it. When we run this enrichment analysis and correct for multiple comparisons, two things survive: drought recovery and response to Caracan. We were really excited to see both of these. This is not taking drought into our priors. We weren't looking for drought. We were just looking for what's turning over fast. And at an 800 kilometer scale, the extreme turnover loci are associated with drought recovery. Caracan is exciting too. It relates to a parasitic plant that's important in sorghum, but alas, not in the scope of this talk. So what we have from here is a non-targeted scan for allelic differentiation finds drought. We didn't have to be looking for drought to find drought. That's exciting and promising, and it's showing us at what scale drought adaptation is happening to help us think in future about where to draw the lines. keen-eyed listeners might have noticed, I haven't actually used the pangenome at all. I have snow-jobbed you in with the idea that this was going to be a pangenome talk and not talked about pangenomes. So pangenomes are really exciting and important because reference-based approaches to calling genetic variation do not necessarily capture structural variants. If something's not in your reference genome, you're not going to get it. So incorporating many genomes together allows us to see that kind of variation. And when we zoom in on this region I've highlighted, it's a tandem array of a gene that I was not previously familiar with, but that's in the lignin deposition pathway. But a tandem array is many copies of the same gene lined up in a row. These are nightmarish for reference-based calling because if the copies are similar to each other, you don't know which copy your reads are aligning to. You're getting spurious signals of heterozygosity. You don't know how your copy numbers are varying across your samples. Having a pangenome lets us start to untangle that. So this is the 30 references in the pangenome, and this is just showing you presence-absence variation relative to the reference we used at the bottom. So I'm running out of time. Let me just shout out the Hudson Alpha Institute for Biotechnology. We build really cool plant genomes, and we love to collaborate. We are full-time researchers. We are an independent, nonprofit research institute. This is what we do, and we love to collaborate. And the JGI, the Joint Genome Institute in the Department of Energy, funds a lot of basic science and genomic research. So there are rolling calls for investigators. Come talk to me about either of these things, and I might have time for a question. Yeah.
I'm interested in wavelet transformation and wondering if it's picking up loci that other GEA approaches are not.
I don't know how extensively it's been competed against other GEA approaches. I know in the paper that it's picking up different things at different scales, which I think is a real strength of it. And like, one of the things that's nice about it as a genome environment diversity approach is you might not know a priori which environmental variables are important, so it might be drawing your attention to things that are happening at scales you weren't considering. Very happy to talk about that more. Jesse had a great example of like mapping different kinds of environmental variation onto the continent to point out how like different variation is happening in different scales in different places. Excellent. If there are no more questions, thank you again for coming, and I'm back in front of some collaborators again.
My name is Swapna Subramanian. I'm a PhD candidate in Dan Bolnick's lab at the University of Connecticut, and today I'm going to be talking about yeast adaptation and maladaptation and community dynamics in Connecticut apple orchards and apple varieties. So to start off my talk, I have a quote from the icon Charlie XCX. In her recent song Apple in the album Brat, she says, "I think the apple is rotten right to the core from all the things passed down from all the apples coming before." And before I talk about how this is relevant to my talk, I have an important note, which is that there is a TikTok dance associated with this song, and Dr. Daniel Bolnick has made a deal that if I get a standing ovation at the end, he will do the dance. So I hope we're all on the same page about what's happening here. But this is extremely relevant to my research because while Charlie is talking about the psychological ramifications of being a woman of Indian descent, the things passed down from apples that are rotting them are, in fact, wild yeast and bacteria, but we're talking about yeast today. And yeast is a really common model organism. Scientists have been studying yeast for over a century, and humans have been interacting with yeast for even longer than that, and yet little is still known about wild yeast ecology and evolution. We do know some stuff about yeast in the context of economically important stuff like alcohol and food, but we still don't know a lot about wild yeast in more natural contexts. So this brings me to where I study wild yeast, Connecticut apple orchards. So I go to Yukon, which is in Connecticut, and one of the biggest things to do there is go to apple orchards in the fall and go apple picking. And just around Yukon, when I Google Maps it, there's 20 apple orchards, but that's just the max that Google shows, and there's more than 70 apple orchards in Connecticut alone. So you can imagine this is a huge industry in Connecticut. And as I was going apple picking, I started to think of apple orchards as evolution experiments. So let me explain. Some of these orchards are generational and have been operating for hundreds of years, with Rogers Orchard as an example having been open since 1809. And so coming from a stickleback lab, always looking for natural evolution experiments, I started thinking about how a lot of the same varieties are grown in these different orchards and subject to different treatments. And these clonal apple varieties are almost like replicated habitats across these orchards. And this led me to think that this is probably the perfect place to study yeast evolution in nature. And you might be wondering, where are you getting yeast from apples? Because on the skin of apples, it's really hard to isolate yeast, and it's just hard to isolate wild yeast from bark and most surfaces. So I actually ended up looking below the trees. And below the trees are apples that have fallen, gotten punctured, and are open for yeast activity because the yeast loves sugar. And not only do the yeast love sugar, but lots of bugs love sugar, and so they're also going from apple to apple, carrying yeast on their legs, and there's lots of Drosophila there, so any Drosophila people, if you want to do a coevolution experiment, hit me up. But these rotting apples are almost certainly sources of yeast evolution activity. And we are saying almost certainly because that's what we're answering here today. Are they evolving in these apple orchards? And so to answer this question, first I'm going to characterize the wild yeast community composition in these orchards and varieties, because what yeast are even there? We don't know. Next, I want to see if these yeast communities are functionally different by characterizing their performance in apples. And then I'm going to zoom in on a focal species to see if these yeast are evolving and if they're locally adapted to these apple orchards and apple varieties. And then finally, I'm going to take it back to the communities and determine whether the evolution of this focal species is affecting community composition and community performance. So let's address this first question about wild yeast community composition. So to do this, I visited 8 orchards and collected rotting apples from 3 varieties, which the apple orchard owners were really weirded out by me, but it was okay. And I cultured yeast from those rotting apples, isolated 8 colonies from them, and I used ITS region barcode sequences to characterize the fungal microbiome. So our yeast community composition in these orchards and varieties looks a bit crazy. You can try and find your favorite yeast species on the side here, but the one that I determined was my favorite was the one that's the most abundant, because I thought that this would be the perfect focal species to zoom in on later. But when it comes to actually answering the question of whether yeast community composition is different between orchards and varieties, our diversity statistics can show us something with Shannon alpha and Simpson alpha is what I'm showing you here. And so based on this, we can say that our alpha diversity, so species richness, is significantly different based on orchard with both Shannon and Simpson. But by variety, we aren't seeing significance, which makes some sense because they are clonal and are replicated across these orchards, but they're still like the same thing. And with the Simpson alpha, we do see that orchard and variety shows significant interaction, but we aren't sure what this actually means in terms of their function in these apples, and so I'm going to address that next with the yeast community performance. And so I tested this by taking those same yeasts that I isolated, pooling them into communities at equal frequency, and then doing a full factorial transplant experiment by stabbing apples, injecting them with yeast, and then allowing them to rot for 7 days. And then I used that rot measurement as a measure of their performance. And so before I get into the results of this, I'm going to show you a little example with red apple and yellow apple. And so all my experiments started off like this with both yellow apple and red apple getting yeast from each other. And they start off at the same size, and then after they rot for 7 days, it didn't look this pretty. It looked a little bit horrific. But, we see that the yellow yeast did really well in the yellow apple, and the red yeast also did really well in the red apple. So what will this look like on my figures? I have rot size when yeast is native on the x-axis and when it's non-native on the y-axis, and the line of the diagonal line is if they're equal. And so since the yellow yeast does best at home, it's on that side of the line near where the rot size when yeast is native on the x-axis. And you can see that yellow does better at home, red does better away. So let's get to the actual data. When looking at yeast community performance across different orchards, we see a lot of variation. So there are some yeast communities that do better at home, and there's some that do better away, but they are doing significantly different than each other. And when you look at variety, you also see the same thing, which was pretty cool because the effect of variety is different from the effect of orchard. And so these varieties, even though they're the same, replicated across the different orchards, we are seeing some similarities in the varieties across the orchards. And so now we know that these communities are different and they're actually functionally different too. So let's zoom in and see if they're evolving and see if HURing specifically is evolving. And so to do this, I took all those yeast that I isolated and identified HURum samples from them, and again did a full factorial transplant experiment. And this time it's a little bit of a messier graph because all of these points are different strains of yeast of HURum. But you can still see that there's grouping based on orchard, and specifically that Dondero orchard seems to be really maladapted. But you can see that there's variation in these populations too, which is pretty fun for evolution purposes. And when you look at variety, you again see something similar where there's a little bit of grouping, but what's really interesting is that Cortland yeast seem to be locally adapted, and Golden Delicious yeast seem to be locally maladapted. But this is super exciting stuff because this means that there's probably evolution happening, and it might be parallel on the level of variety and also on the level of orchard. And so HURM genomic data is going to be coming soon, and we can actually see if this is reflected in their genomes. And so finally, let's take it back to the communities and see if the evolution of this focal species is affecting community performance and community composition. So first, for community composition, on the y-axis here, I have log-fold change in fitness when native versus non-native as a measure of local adaptation. And so you can see that those populations of HURM that are more locally adapted generally have higher proportions of HURM in the community, but this is not significant. And next, whether HURM local adaptation affects community performance, we also see that this is not significantly correlated. Sometimes both the population and the community both do better away. Sometimes they both are better at home. And sometimes they aren't in the same direction at all. And so there seems to be more going on in these communities and with the species interactions than can be found with just zooming in on this one really abundant species, even though it is super abundant. So what conclusions can we draw here? You guys are stuck until 5:30, so. First, does wild yeast community composition vary between orchards and varieties? Yes. And also, yes, their performance varies. That focal species of yeast, HURM, does seem to be locally adapted and sometimes locally maladapted to different apple orchards and varieties. And finally, the local adaptation of this focal species does not seem to be driving the community composition and performance. But what's really exciting about this is that it's almost a new system to study the repeatability of evolution, and I'm going to be addressing those questions, and so get excited. So finally, I'd like to thank the Bolnick Lab, the JRFP for funding me, and Connecticut Orchards, and feel free to scan this QR code, especially if you want to get the video of Dan dancing afterwards. And thank you all very, very much. Someone has to stand up, just one person. Oh, she's standing up. Okay, wow. Oh my God, wow. Thank you, thank you, thank you, thank you, thank you, thank you, thank you. I'm so glad you guys all love the talk so much. Any questions? I know we all ran out of time, but yes.
This is an ecology question, and I'm not an ecologist, but how does the community composition work? Like, are you interested in finding out in what order the yeast colonize, and do they, like, make the environment good for other yeasts? Is the really abundant one, like, a late-stage succession yeast? Tell me about ecology.
These are such good questions, and definitely worth exploring. And I don't know the answers to them right now, and I don't know if I will know the answers to them during my PhD. But I think that there's definitely really interesting dynamics happening on these running apples, like on this tiny scale, and actually testing whether succession plays a role and what these species are doing when they're all sitting in this apple eating sugar is super interesting and worth exploring. But I don't have answers yet. Yes.
Yeah, I also have an ecology adjacent question.
Okay.
So how do you, or maybe this isn't important, but how do you disentangle, you're collecting fallen apples, how do you disentangle the yeast being in the apple orchard and the yeast being just on the ground or in the site? And I'm wondering because, you know, in the whole process of apples and selling apples, there's different sort of complications with that, right? They might harvest and pick apples and send somewhere else where there's other yeast samples that are different, have different adaptations. So
Definitely.
yeah.
Yeah, so when I go and I collect like directly on site, like I go right below the tree that is the variety that it is, and I grab an apple, and it really is possible that they're coming from the soil, but at the end of the day, like when they're spraying, they aren't spraying just the trees or just the apples, they're spraying everything, right? And so the method by which the yeast colonize the apples, to me, isn't as interesting as the fact that they're there, you know? So like the fact that they're there means that they're going through these dynamics in these apples and like being distributed by these insects, and they could be coming from the soil, they could be coming from insects, they could be coming from the air. That's the beauty of yeast, so. Yes, Mark.
So do you have any idea of chemical ecology among varieties or among orchards, especially among orchards? Like what's different?
Great question. Okay, so among varieties, there's a lot of like already established like differences in terms of like cell structure, sugar content, like nutrient content. In terms of the orchards owners like hate telling me what they do. So they don't want to tell me like, oh, we spray this at this time, like any of that. So I have been establishing relationships with them for like the past 4 years of my PhD. So I'm hoping that eventually they tell me, and if they don't tell me, I'm hoping that I can grab some leads and do some testing. But that would be like really good to tie into this to actually see if that's what's driving this variation that we see in adaptation and application. Yes.
Yeah, how many strains of yeast are found on like each fruit? Like, can you guarantee that you're inoculating everything with the same strains of yeast? Or can that be where a lot of your variation comes from?
Wait, I don't know if I understood your question.
So within species, there's a bunch of different strains that can behave really drastically different.
Yes.
So are there more than one strain found on the apple? And are you basically isolating one strain off of the apple and then inoculating them all with the same one? And then, like, yeah, how's that
So the way that I did the different strains was I first isolated 8 colonies that are definitely isolates from the rotting apples, and I used barcoding to identify H. uvarum. So I don't know whether, and then I tested 5 strains from each orchard variety combination, and so I can't be sure until I do genomic sequencing whether they're like truly genetically different, but the ones from different orchards, I'm almost certain are genetically different. But again, I'm not going to like make any promises until later. But it could be that there's plastic variation in one strain that I isolated, and I just isolated the same strain over and over again. But I highly doubt that, especially because they're from these geographically distant orchards, and within orchards maybe it's more likely because everybody